British Science Week is a 10-day celebration of science, technology, engineering and maths. And the theme for 2021 is Innovating for the Future. In this video, we will hear from lead osteologist Lauren McIntyre about some of the innovative scientific techniques her team will be using to analyse burials recovered from our archaeological excavations at Trinity Burial Ground in Hull.
One of the scientific techniques that we're going to be undertaking is ancient DNA analysis. Now, this is not something that we routinely do with archaeological assemblages, but we thought it was appropriate to apply here at Hull Trinity. One of the things we're going to be using DNA for is investigating family relationships. So in the Victorian period, people tend to bury their dead in stacked graves, which means that we have multiple individuals buried on top of each other. We'll be using DNA to investigate whether these stacks contain members of the same family. So for example, we might see if we have parents and children, or if we have grandparents and children, or any other sorts of relationships. We can also use DNA to look at the sex of juveniles, so of child skeletons, because we can't actually tell whether juvenile skeleton is male or female just from looking at the bone. With pathogen DNA, we can of course identify diseases that we can see in the skeleton, so things like tuberculosis. We're also seeing a little bit of syphilis. That's a lot more difficult to identify using pathogen DNA, but we can try and look for it. I think interestingly, we know that there were cholera outbreaks in Hull in 1831 and in the late 1840s. So cholera is not something that does leave a physical trace on the bones. We can't see any lesions that were caused by cholera. So that's one of the diseases that we can investigate using bacterial DNA analysis. Another scientific technique that we're going to be applying to the skeletons from Hull Trinity is stable isotopes. Now, stable isotopes are atoms that we can find in your bones and in your teeth. And the ratio of the different types of atoms that are in there are influenced by what people have been eating and drinking. So, for example, rock and soil, so geology and soil, contains different amounts of different types of isotope. When plants are grown on this soil, then the plants take up the same isotope signature as is found in the soil that they're grown in. So then when animals eat these plants, they also take up that same isotope signature. And then if humans eat the animals, then humans also copy that signature. So we can see there's like a chain where this isotope signature is being copied between geology and between the humans that are consuming things related to that geology. We can use these isotopes to determine what sort of things people have been eating. So for example, whether they've had a lot of certain types of plants in their diet, or maybe they've been eating a lot of food that's come out of the sea, or whether they're eating animals that have been roaming around on the ground, so terrestrial animals. The oxygen signature from water is very much a reflection of like local geology and climate. So we can look at oxygen ratios to see where a person has grown up. And so in the case of Hull, we can look at these things to determine whether somebody grew up locally to Hull or whether they grew up further away, maybe elsewhere in the UK or even abroad, and then they came to Hull and died here and ended up in the cemetery. A third technique that we'll be using is incremental dentine analysis. Now, this also uses stable isotopes, but in a slightly different way. So we're actually going to be using it to look at weaning patterns in very young individuals. So assuming that during the 19th century, mothers were giving their babies breast milk, obviously at some point they would be weaned onto different types of food. And this, we can see this dietary change in the isotopic signature in the teeth. So the way that incremental dentine analysis works is that we look at different parts of the tooth because we can see when those different parts of the tooth grew. So if you look very, very closely at the surface of a tooth, you can see small, almost rings, a little bit like tree rings. So where tree rings show you how much a tree has grown over a certain period of time, we can do the same thing with a tooth, but looking at the tooth enamel, and we can see how much a tooth has grown over a certain period. Now, if we compare different parts of these tooth enamel rings, then we can see changes in the isotopic signature.